Hello, and welcome back to the lecture on Chapter 5, Metabolism. We had just finished looking at an overview of metabolism, how catabolic reactions release energy and release metabolites so that we can perform anabolic reactions using the energy in ATP and those individual building blocks to build larger macromolecules, and then how this continues recycling. In part 2, we are going to look at a little bit more detail about how those reactions are done and importantly, the role that enzymes play. So the basic kinds of reactions, many of the basic kinds of reactions that we're talking about are referred to as oxidation and reduction reactions, or ox-redox reactions, redox reactions. In a nutshell, we're taking electrons from one molecule and transferring them to another molecule. So the electron donor does what you might expect, gives up the electrons. And then the electron acceptor accepts the electrons. The terms are confusing. Let me just warn you about that. Yeah, electron donor and electron acceptor, easy, but who's getting oxidized, who's getting reduced? So these reactions have to happen at the same time, obviously. If you've got one thing, one electron or two electrons, it's got to be transferred. So one of them's giving it up, the other one's taking it. The electron acceptor is often what we refer to as an electron carrier. It is going to take those electrons and put them somewhere else. The carrier is often the hydrogen ion that's at the end of one of these molecules. And there are three important electron carriers. You may remember these from your biology classes. NAD plus can become NADH. It's an electron carrier. It carries the electrons over to the electron transport chain. FAD, maybe FADH2, you might remember that one. Anyway, these are electron carriers that we're going to deal with. And the simplest diagram of this, an oxidation reduction reaction, a redox reaction. Yeah, it's as simple as this. There's an electron donor, has the electron, and it transfers it to the electron acceptor. This is where the terms get confusing. If you can remember that the electrical charge on an electron is negative, so the overall charge on the electron acceptor is going to go down, we say the acceptor gets reduced. And what's confusing about that is that, yeah, it's gaining an electron, but its electric charge is becoming more negative. And so that's where the term reduction comes from. And then the other term that's a little bit confusing, oxidized, there might not be oxygen involved, but the reason it's called this is oxygen is really good at taking electrons from things. And so we say it's been oxidized, it's been stripped of an electron. So the donor has been oxidized, the acceptor has been reduced. What does this have to do with anything? When we do the catabolic reactions, we're breaking down the molecules and releasing energy, we kind of store up some of that energy into a high energy molecule. And remember, we said we put that phosphate group, that third phosphate group, onto the ATP. That process of putting a phosphate group onto something is referred to as phosphorylation. That means inorganic phosphate, which is a phosphate with 4 oxygens on it. It's added to some molecule. So we take ADP, we phosphorylate it to make it ATP, adenosine triphosphate. In the first few reactions, when we think about metabolism, you think about, you go through glycolysis and then go through the Krebs cycle. When we form a few ATP molecules during those series of reactions, that's referred to as substrate level phosphorylation. there's an enzyme involved and it's helping us to put this phosphate group directly onto another molecule, so substrate. In contrast, when we say oxidative phosphorylation, that refers to what's happening when we do cellular respiration. Remember the electron transport chain, and then pumping protons across the membrane, and then the protons are allowed to come pouring through another enzyme called ATP synthase that moves in response to this, chemiosmosis, the protons pouring down in through that enzyme. Fantastic stuff. That's oxidative phosphorylation. And then finally, just for completeness, remember I said that some organisms can get their energy not from molecules, but from actual sunlight, from photons. It's kind of a similar process. Like we're using this energy from the sun to just put a phosphate group onto something. And so photosynthesis is an example of photophosphorylation. And then all of these ATP molecules that have been made through these different processes, the whole reason we use those is to pay for stuff. So the anabolic pathways where we're building up larger molecules like DNA or proteins, they're made by using that energy of ATP, by breaking that third phosphate group off. And we have large molecules that help us with those reactions, and those large molecules are referred to as enzymes. They're large proteins that help the reactions go. So I mentioned this before, they're just catalysts. They just make the reaction go faster, a reaction that might happen anyway, but these molecules allow that reaction to happen more readily. I think of them as like matchmakers. Like these two people might have eventually gotten together, but if I'm gonna like invite people out, let's go to a restaurant, I'm gonna sit these two people next to each other because I think they're gonna get along really well. That's what enzymes do. Increases the likelihood of a reaction. And then what I say by organic catalyst, organic, remember that just means that the molecule contains carbon. So for example, proteins. These are enzymes that catalyze reaction. So these are proteins that allow reactions to go faster. That's what enzymes are. There's a bunch of different categories of enzymes. I'm not going to go into a huge amount of detail with it. What I want you to notice is the ending of the names ASES, ACEs, ACE reductase, transferase. That's the convention for naming enzymes. So if you see a molecule and it ends in ASE, that's likely an enzyme. I'll give you examples of ones that you've already seen before. So these are the 6 different categories. Hydrolysis, so they do hydrolysis reactions, breaking things down. We saw this when we were talking about taking amino acids away from each other, apart from each other, taking sugars apart from each other. These are hydrolysis reactions. Examples include an enzyme called lipase, used for breaking down lipids, proteases for breaking down proteins, amylases for breaking down carbohydrates. We've got plenty of them for our digestion, and microbes have some as well. Ligate polymerases. You probably remember this from your biology class. There's a polymerase, or DNA polymerase, RNA polymerase. These are the enzymes that allow us to put individual nucleotides together to form longer nucleic acids. And let's see where we're going to see kinases, transferases. You may remember kinases. Well, doesn't matter. There's lots of different enzymes. They have different jobs. What they all have in common is they're made out of protein, and their job is to help us do a reaction, either put things together or take things apart. All right, and that's the end of part 2. See you over in part 3.